Well, because we know that age is the main risk factor for many neurodegenerative diseases, and we also know that one of the earliest and most consistent changes that occur with age is in the brain vasculature. The blood-brain barrier, which is a specialized structure that exists in the brain blood vessels and that protects the brain from harmful substances coming from the periphery, starts to weaken. And importantly, this happens long before any cognitive decline becomes apparent. But what we don't know, or we didn't know, is if there are some regions that are more vulnerable to this aging process than others. Because if certain regions of the brain are more vulnerable to vascular aging, that might explain why there are some regions that are the first to show a decline in disease. And understanding these early changes at the blood-brain barrier can give us new clues about disease vulnerability and potentially point to ways we could protect the brain as it ages. We use young and aged mice of between 2 and 4 months and 18-20 months old. We use both sex to account for potential sex difference, and we focus on 3 brain regions: the prefrontal cortex, the corpus callosum, and the hippocampus. To study the different components of the blood-brain barrier, we use immunofluorescence and electron microscopy. Aging does not produce uniform changes across the brain vasculature. Instead, the prefrontal cortex emerges as the most affected region. We observed an increase in transcellular permeability markers in both females and males. We also detected a thickening of the basement membrane, but specifically between endothelial cells and astrocyte endfoot. Interestingly, while the molecular markers we assess show little change, astrocyte endfoot themselves became significantly enlarged, increasing in size by around 40%. The hippocampus, in contrast, showed only limited alterations, a subtle increase in transcellular permeability markers that was restricted to females, and an increase in the astrocyte endfoot protein beta-dystroglycan. The corpus callosum, somewhat unexpectedly, showed no significant age-related changes at this stage. Our results show that aging doesn't affect uniformly the blood-brain barrier across the brain regions. The prefrontal cortex appeared as a particularly sensitive structure to aging, showing structural changes that could make it more vulnerable to age-related conditions. On the other hand, the corpus callosum and the hippocampus seemed relatively resilient at this stage. And overall, I would say that our results highlight that early vascular changes are region-specific and could help us understand why there are some regions that are the first to show dysfunction in disease.